Welcome to the Centers of Excellence in Hematology seminar series on Flow Fridays. Today, we're pleased to hear from Dr. Pratibha Singh, who's going to talk about flow cytometry sample preparation and staining. These presentations are recorded and will be made available on our U24 CCEH YouTube site, but also you can find them on our website, which is cceh.io. So without further ado, let's get started. And thank you again for your taking the time to present to us today.
Thank you so much, Dan. And thank you so much for helping me with this, all the technical issues and then and all the issues that we are coming through. So today, good afternoon, everybody. Today, I will be talking about the flow cytometry sample preparation and staining. Flow cytometry. What is flow cytometry? Flow cytometry, it is the measurement of cells in a flow or stream. So this is a laser-based tools that measures the biological and chemical properties of the individual cells or particles that are flowing in the stream of fluid. That is flow cytometry. When we are measuring the cells that are in cell suspension and when it is passing through the machine, it is in fluid. That is flow cytometry. For any flow cytometry experiments, that this is our basic principle of flow cytometry. First, we prepare the samples, biological samples. That can be biological samples, can be tissues, tumor tissues, blood samples, other biological fluid where we can get the cells. We label the cells with the fluorochrome conjugated antibodies or other reagents. And then we run these samples, fluorochrome conjugated antibody labeled samples to flow cytometry machines. In the flow cytometry machines, the cells are in linear fluid stream that move through a focused light source, and the light source is the laser beam. When the fluorochrome conjugated reagents get activated by laser beam, it emits light of a larger wavelength, and that wavelength is filtered by mirrors and then detected by sensitive light detector that includes the photomultiplier tubes. And this fluorescent light is later on converted to digital signal that we measure in the form of cell events. This slide shows the basic outline of sample preparation and staining for the flow cytometry analysis. So as I said, the samples can be a cell that are like blood samples or any cells from other tissues. We first prepare the single cell suspension. That is a very important step for the flow cytometric analysis. And when we get the single cell suspension, we label the cells, we add the antibodies that are fluorochrome conjugated or other fluorochrome conjugated reagents that can bind to the specific marker on the cell surface. And then we incubate the samples in dark at 4 degrees on ice, and after 30 minutes, normally after 30 minutes, we wash the cells and we add a staining buffer, centrifuge, and discard the supernatant and add the staining buffer and acquire the data. But if we can't do the acquisition on the same date, we should fix the cells and then fix the cells with the paraformaldehyde, normally 1% paraformaldehyde, and then acquire on next day. So this is a basic outline of sample preparation and staining. It includes first single cell suspension, adding antibodies, incubation, then washing and resuspension and fixation, and data acquisition. So the key feature that for the good sample preparation, that includes the sample should be in a very good quality, because it is crucial. It will give you high quality data, and data will be reproducible. So there are two important things that we should pay attention. We get samples. First, the sample should have a high cell viability. Viability should be very good, and the sample should be in single cell suspension. Why viability is important? Because the dead cells that can non-specifically bind to the antibodies that we add to identify the markers, those antibodies can non-specifically bind to the dead cells, and that can lead to false, the high background false positive for the cell surface and intracellular markers. So cell viability, when we want to do flow cytometry staining, we should make sure that samples have good cell viability. More than 80–90%, it is very good. It is important to have that kind of good viability. And then also samples should be in single cell suspension, because when samples are not in good single cell suspension, it can the big chunk of sample of the cell clumps can clog the flow cytometry machine, and it can also create cell doublets. Cell doublets means that the cells clump together, and so this that can reduce the number of actual single cells, and so it can give which can like decrease the accuracy of the compromise the accuracy of the signals that we get. So it is important to have single cell suspension and the good cell viability. These are two important things that we should consider when we do any kind of flow cytometry analysis. To how we can do that, how we can improve the cell, how we can get good quality sample. So first thing, make sure get cell samples from fresh. The sample should be fresh, and sample preparation and staining should be finished as soon as possible. Although in like some condition, some cases it is not feasible, especially in clinical samples. There we should like standardize and to determine that how long we can wait before we can wait before we do the staining and get the good data. For the critical point for blood samples, because the blood samples are the sample of choice for clinical application. So we should consider that what kind of anticoagulant we are using, because to stain blood samples, the blood samples should not be clotted. So we should use anticoagulant, and there are several anticoagulants that can be used, including EDTA, sodium heparin, acid citrate, dextrose. These are anticoagulant, but these anticoagulant can impact the ability of antibodies, fluorochrome conjugated antibodies, binding ability to the markers. So it is important to when you are doing any experiment, you evaluate that which anticoagulant you should use for your study. And normally I use EDTA and it works for me, but the markers that I use for flow cytometry, but there is a possibility that some markers, like I have heard that CD11B expression may compromise when you use EDTA. So you should try when you're doing experiments, before doing the real experiment, you should check that which anticoagulant you should use for your blood sample. And sometimes you can use the whole blood sample for flow cytometric analysis, just do RBC lysis and then stain. But sometimes when you want to see the rare population, we need to remove the granulocytes, RBCs to increase the number of cells of interest. In that case, we use FICOL. So FICOL gives that monocyte, and so when you want to use FICOL based monocyte enrichment, we should do that also as soon as possible when we get the sample, because the delayed processing of whole blood sample can result cell loss, as well as it can cause granulocyte contamination into mononuclear cell layer. So the bottom line here that when we get blood samples or any samples, we should try to get fresh sample, try to do as soon as possible both the sample preparation and staining. For the solid tissues, like any like tumors, tumor tissues or any other solid tissues, this is important to make it single cell suspension. And there are several reagents that those are available, commercially available, that can be used. That including that enzymatic digestion, mechanical and chemical single cell suspension. And these reagents that we can use, or mechanical or chemical dispersion, we can do a pilot experiment, and then see that which one is the best for in our condition, which one gives us better data. So this is when you are starting your experiment, you should do a pilot experiment and check which one works best for you. For my case, I do bone marrow cells and also spleen cells. I use collagenase type 1 and based digestion. For spleen tissues, even when I want to isolate mesenchymal stem cells, endothelial cells from bone marrow, mouse bone marrow, I use collagenase type 1, and that works for me. And mechanical separation, of including vessel, motor and vessel, can be also used, and then filter the cells. And that is when you prepare the suspension, if the cells should not, we should count the cells, and the cells number should not be less than 100 million cells per ml. So once you have the samples, and you want to do the staining, it is important to use the right type of tubes, the cell concentration and the sample volumes. These things are important when you want to start your staining. So the sample tubes that for a normal flow cytometric analysis that I do my lab does. We use 5 ml polystyrene tubes. We get that from Falcon brand. Other brands also can, you can purchase from other companies, vendors, but this one works very well for us. And so 5 ml polystyrene tube, it is very good. For LSR2, I normally use LSR2 machine, and then that is also good for other flow cytometry. When we are using biological samples like human samples, it is very important to use tubes with cap. We should be very careful when we are handling human samples. Even mouse sample also, we should try to use tubes with caps. Cell concentration, that when you are ready to stain the cells, the final concentration should be minimum of 1 million cells per ml. if we want to it is the minimum number of cells that you should have, and so that that you can use for phenotypic analysis, apoptosis, DNA content, and similar other experiments. The volume, normally we use 250 to 300 microliters of volume for when we run the samples, and the cell concentration that we use, it is 1 to 5 million cells per ml. So 250 to 300 microliters concentration, 1 to 5 million cells per ml. So now we have prepared the samples. Now we are ready to stain the cells. So these are the key factors that we should consider when we are ready for staining. The one is absolute cell count. How many cells should be 1 to 5 million cells per ml concentration. What kind of a staining buffer we are using, elimination of aggregates and clumping. The controls for flow cytometric analysis, controls are very important. So there are several controls that I will be talking soon, that we should use elimination of dead cells from the analysis and fixing cells with fixative. Normally it is 1% PFA, paraformaldehyde. So the staining buffer that normally there are several vendors that sell the staining buffers, and you can also prepare by yourself. The flow cytometry staining buffer is a buffer solution that contains the fetal bovine serum and sodium azide. Sodium azide works as a preservative, and it can also inhibit the capping of cell surface antigen. This buffer can be used for antibody and cell dilution step, as well as washing step. For all the this buffer can be used throughout your staining process. That include that preparing the antibodies, when you are diluting the cells, washing washing the cell surface, even for the intracellular staining. There are, for normal situation, this normal buffer, staining buffer, that has some protein like fetal bovine serum or BSA that prevents that non-specific binding also, and sodium azide. But for a special condition, like when you have more than one brilliant violet conjugated antibody or super bright conjugated antibody, then it is recommended to use a specialized staining buffers that are available from Thermo Fisher's, eBioscience. We can purchase and use that to improve the sensitivity of brilliant violet fluorochromes. It normally, if we have only one brilliant violet fluorochrome, it is not required. But if we have more than one, it is recommended to use a specialized staining buffers. The elimination of aggregates and clumping, or although it is like we can like when we prepare the single cell suspension, that time we should try to use filters and syringes and the needles. We can eliminate, we can reduce the clumping aggregates. The cell types like monocytes, granulocytes, adherent cells, they have tendency to aggregate, and it can clog the instrument. If they are they are big, they can clog the instrument, and it takes time to clean the probe there, and then it can affect your data acquisition. So if you see your sample, and if it has clumps, visible clumps, or a strand of cells in your sample tube, it means that there is some aggregation. There is some aggregates or like the samples are not dissociated properly. So if you find that the cell suspension should be filtered with 20 to 30 micron filters, and then to remove the aggregates before running the sample, or like when you're preparing the samples in the beginning, you should filter the samples. But when you have samples ready, when you have stained the cells, and then or even the process of staining the cells, and if you see the aggregates, clumps, so then you should filter the cells with 20 to 30 micron filters. And if you see the large debris, then you should use 70 micron filters. In addition, that I normally use 20 and 30 micron filters to remove the aggregates. In addition to that, if in your samples, if there is a lot of cell death, if you know that the cell viability is not very good, and then you will most likely get a lot of aggregates. And what happens that the DNA that released from the dead cells, that can contribute to cell aggregation. So we should use DNAs. DNAs that DNA should be added to the all sample preparation steps, including the wash step. What it will do, that it will eliminate the DNA, and it will actually, DNA is an enzyme that degrades the DNA, so it will eliminate the free DNA from your sample, and you reduce the chance of cell clumping and aggregation. There are like also commercially available products such as Ecumex from Invitrogen that can also be used to keep the cells from clumping. So once I've talked about the staining buffer and then how we can eliminate the cell clumping and aggregation, and also like it is the next step is to make the proper controls for your flow cytometric and staining. So when we make the controls, we should at least make these 4 controls. These are very crucial. The one is biological control, that are normally the unstained control samples. This can help to identify the autofluorescence that present in your sample. Compensation control, isotype controls, and fluorescence -1 controls. I will talk a little bit more about these controls just in a couple of minutes. So we have removed the we have tried to eliminate the cell aggregation, and then we have also the controls. Then it is like how there are, even it doesn't matter how good our samples are, but still we will have some dead cells. So these dead cells are more autofluorescent than live cells, because the antibodies that we add to detect the specific marker can bind non-specifically to the dead cells, and it is very challenging to remove the dead cells just simply based on forward and side scattered. So it is always recommended to use a viability dye, fluorescence conjugated viability dyes should be added in your sample preparation to exclude the dead cells from flow cytometric analysis. There are 2 types of viability dyes that are available. One is the DNA staining dye, and second one is amine reactive dyes. So when cells are dying So what happens that the cell membrane integrity compromise. So when you add DNA staining dye, means like that the DNA staining dye will go inside the cells, and because the membrane is compromised, and it will bind to the DNA. And so that way it will exclude the dead cells. So DNA staining dye includes propydium iodide, hashed, these and also 7-AD. These are the dyes that are commercially available, excited by different natures, and just like propydium iodide, excited by I think that 488, and hashed, it is violet laser. And the amine reactive dyes. So this is another type of dyes that can be used to exclude the dead cells. So in healthy cells, there are fewer amine group that are present on the cell surface, but when the cell are dying, their membrane integrity is compromised. So and there are a lot of amine groups that are present inside the cells. So when we add amine reactive dyes, they go inside the cells and bind to the intracellular amine group, and that are more abundant in the cytoplasm than extracellular amine that are present on the cell surface of live cell. So that based on that, we can also exclude the dead cells. In my lab, we use amine reactive dyes on regular basis. This one, for the amine reactive dyes, we first add this dye, and then after we add all the antibodies. What the DNA staining dye, normally people first add all the antibodies, stain the cells, and then just before acquiring the data, add propydium iodide or hashed. So both are good, and just based on that, how many fluorochromes you have, how many antibodies you are using, and which channels are available for your experiment, you can choose one of the dye. And it is very important to exclude the dead cells. Simple forward scattered and side scattered is not sufficient to exclude the dead cells. This is the outline of simple cell surface marker staining protocol for flow cytometry. So the first is I just talked, that prepare the cell. So get the sample, try to do as soon as possible, collect the cells, filter the cells through 20 to 30 micron filters, centrifuge it, and discard the supernatant. Then add the staining buffer that commercially available. If you don't have commercial available staining buffer, you can also prepare the staining buffer by adding 0.1% BSA or 2% fetal bovine serum in PBS, and you can use that as a staining buffer. So after once you discard the supernatant, add the staining buffer to the samples, and then count the cells. You can use the simple hemocytometer, or you can use a HESCA, HemoVet, depending upon what type of samples. So what is the origin of your cell? You can use either manual or automated cell counter, and adjust the cell concentration around 10 million cells per ml. Then set the samples and controls. So for the controls, there are 5 these controls are very important to do the flow cytometry analysis for staining, and then analysis, and then samples. So samples, if we have, just like depending upon how many total biological samples we have, 5, 10, 15, 20, so those many samples. So the controls, so first control is the compensation control. So the compensation control, this is a single color stained control used to correct for spectral overlap between fluorophores, and it is necessary for any flow cytometry studies. So what does it mean? That when we are using multiple fluorochrome conjugated antibodies in one tube, so there is antibodies that are like when they excited by a specific wavelength and then emitted the specific spectrum. So what happens that it is not only excited by one channel, but it also, it is not emitted just one specific wavelength, but it also emit it also imitate fluorescence in other channel. So we want to subtract the values that are not belong to that particular fluorochrome. So if we have suppose if we have 6 fluorochrome conjugated antibodies that we are using, so we need 6 compensation control, 1 for each fluorochrome. So and the key feature for the compensation control, so the reagent, the antibody that we are using for the compensation control and the experimental sample should be from the same vial. And if the compensation control and the experimental sample should be treated the same way. The autofluorescence of the compensation control must be identical for both the negative and positive cells. Each compensation control positive population must be at least as bright as the signal that we get from the experimental sample. So the brightness should be either identical or it should be higher than our experimental sample. And it is also important to collect enough number of events when we collect the data to ensure that we have enough events to estimate the mean fluorescent intensity in all the channels as well as for the negative population. So these are the 4 criteria that we should consider when we are using the when we are making our compensation controls. So for each antibody, we should have one control. So if we have 6 antibodies that we are using in our samples for our study, so we should have 6 compensation controls. Another important control is biological control, and that is also called an unstained control sample, because the unstained control samples, it is what happened that the biological samples have the autofluorescence, and some has higher, some has lower. The reason behind that, because there are like some metabolic activities, there are riboflavin, NADP, those things can contribute for autofluorescence. So when we, and that can affect the overall fluorescence of the sample. So it is very important to we should use the same biological sample, same biological that are unstained as a control for our study. Isotype controls, we should also include isotype controls. What are the isotype controls? So isotype controls help to determine the non-specific binding that presents on the cells due to non-specific binding of antibodies to the FC component. So what happens that When we use the fluorochrome conjugated antibodies or any other fluorochrome conjugated reagent, so antibodies especially, they are very specific to specific antigen, and that antibodies bind to the antigen binds to this portion of antibodies. These are FAB, that antibody binding portion. But there is a constant region, that is FC region, that is also that is present on the antibody. This FC region can bind to the FCR receptors that present on the cell surface of several cell types, primarily myeloid origin cell, and then binding of this region to the cell surface FCR can give us false positive. So to make sure that how much a non-specific binding that we getting due to this FC region, we should use isotype control. And so the important point for here, the isotype control and the experimental antibodies, they have the identical FC region. So this region is identical for both, but for the real experimental antibody, this region is very specific to the particular antigen, particular markers. But for the isotype antibody, this is just like very less affinity binding region. So this region, the isotype antibody will not bind to the specific receptor, but it still has this region, and this region can bind to the FCR region that are present on the certain cell types. It makes sure that use the same amount of test and isotype control antibodies. It is also important to, like, if you can buy the isotype antibodies and your experimental antibodies from same company, because sometimes the fluorochrome and the FC ratio, it varies. Different company make when they make antibodies, they have a different ratio. So it is important to get, if it is possible, to your isotype antibodies and the experimental antibodies from same company. The second control is the fluorescent -1 control. So fluorescent -1, what is fluorescent minus? Fluorescent -1 control is a tube of cells stained with all the fluorochromes used in the experiment except one. So suppose if we have 6 colors, the 6 fluorochrome conjugated antibody we plan to use. So in one particular FMO tube, I will have 5 antibodies, but not the 6th one. So this one, why it is important? Because the FMO controls, it determines the cutoff between the background fluorescent and positive populations. So it can give you idea that where I should make our gate, where we should make our gate to make the positive, decide the positive population. If we just have isotype controls and the population is, if the positive population is appears as a smear, so it is very difficult to make the gate just based on isotype control. The FMOs are important. It helps to determine the positive gating and negative gating for the our experimental samples.
Just for clarification, for a naive flow cytometry person, do you run those FMOs every time you run sample sets, or do you set up the program where you know that cutoff will be, and then for future experiments, you can use that? Or is there enough variability in that sort of control background and positive populations?
So it depends, like if some routine antibodies that where I know for sure that I am getting very clear-cut population, and I have done a couple of times, so then I will not use the FMO control for that. But some cell type that if are very rare, and then rare and it is not very clear-cut, so I will try to, I will use FMOs. If it is very crucial for me, and then even if I have used previously, but still I try to add the FMO.
Great, thank you.
So suppose like if it is SLAM LSK, so I know that like it is very clear-cut population, and then I have done couple of times, so then there is no problem. But if suppose in SLAM LSK population, I am looking for a particular marker, and that is not very clean, so then I will use FMO. For ECEH and MSC also, because sometime depending upon the sample processing, that it is not very clear-cut population. So although I have used FMOs for previous samples, but still I will add that. So it just based like how important that molecule is and how rare that population is. So that's the way I do, so this is like as I said that the fluorescence -1, the FMO means like fluorescence -1. So suppose if we have like how many samples here, like, so if we have 4 colors, so here that in particular study, we are using 4 antibodies conjugated with 4 different fluorochromes, FCP, PerCP, and APC. So the FMO control should include that 4 FMOs. So first FMO that should have all the antibodies except FCP. The second FMO, all the 3 antibodies except PE. The third one, all the antibodies except PerCP, and the fourth one, all the antibodies except APC. So that the fluorescence -1. So all the antibodies except, and it is very important to determine the gate. In addition to the isotype control and the unstained biological samples, each controls has their own role. As that the unstained biological sample that can give you idea about the autofluorescence, that isotype control that will give you idea about non-specific binding due to the presence of FCR receptor. Fluorescent -1, it will give the distinct if the population is not clear separation, so it can help to decide the gating, that where our population positive population should be. So when we have all the controls, we have used the viability dyes and excluded dead cells. So the last thing that it is important, it is to use the FCR blocker. As I said that this reason, when we have isotype control, it gives you idea about the non-specific binding due to the presence of this region on the antibody and presence of FCR receptor on the cell surface. So to reduce that, the non-specific binding, we should use FCR blocker. So it is not 100%, but it does significantly reduce the non-specific immunofluorescent staining, the binding. So for the mouse cells, there are several commercially available FCR blockers from different vendors, including eBioscience, BD, Invitrogen, BioLegend. So for mouse cells, there are 60 CD16, CD32 antibody specific FCR FCR2 and 3 can be used to block the non-specific staining. So to do that, what normally I do, or how other people also do, they just add the FCR blocker, normally 0.5 to 1 microgram concentration, and then incubate for 10 to 15 minutes, and then you add your antibodies of interest. For the human and rat samples, people use the purified IgG, Ig from same species or same isotypes as the antibodies used for fluorescent staining, or also use serum. But for human also, there is human specific NTs, CD16, CD32 antibodies are available. So adding FCR blocker can reduce the non-specific bleeding. So once you have all the controls, prepared all the controls, and then you have added like dead cell exclusion dye, you have used FCR blocker, so you have everything now. So then you can add the antibodies according to the recommended concentration. It is just it depends like which where are you getting these antibodies. Yeah, normally we use 2 microgram per million cells, and that works good with us. But the product insert that comes with the antibody, you can follow their instruction and use the recommended amount. And it's also important to pay attention that how many cells you are using, how much volume you have. So based on those things, you should decide that how much antibody you should be used. And then when you add the antibodies, incubate at 4 degrees for 30 minutes in dark, and then when you have done that, then for the acquisition, data acquisition, you should wash the cells with the staining buffer, centrifuge at 300 G 5 minutes, and discard the supernatant. These things is that like the normal, and this is like general protocol. You can change it depending upon the tube size. And we discard the supernatants, and then add 250 to 300 microliters of a staining buffer to resuspend the cells, mix the cells very well with the micropipette, and then acquire the data on flow cytometry. I have talked in detail how in previous lecture that what are the criteria we should use to decide which fluorochrome conjugated antibody we should use, and then what cytometry, flow cytometry machine we should use. First, we should actually, when we are doing these any flow cytometry based experiment, we should check our instrument configuration, which I have talked in detail in previous lecture, but I will record that very soon and then load on and upload on the system. So it is very important to find out that your flow cytometry configuration, how many layers they have, and then based on that, you can decide that how many fluorochrome conjugated antibodies you can use. So those things you should do before doing any study. And for the sample staining, this is general protocol, but it is highly recommended to titrate the antibodies when you are doing first time, when you don't know anything. so you should at least do a pilot experiment to see that that the amount of antibody that you are using, it is giving you good signal and not giving you non-specific staining. So those things are very important to be considered. And here I would like to stop, and please let me know if you have any questions.
Thank you so much for that presentation. If you have questions, please feel free to unmute yourselves. I've also populated a couple of links in the chat for if you want to go learn more about what's available at Indiana's flow cytometry core and some protocols that they've shared. Also, I've populated the upcoming Cooperative Center of Excellence in Hematology workshop that's going to be happening in Indiana. And finally, at the bottom of those chats, I've posted a link to go look at opportunities for funding that are made available through the Cooperative Centers of Excellence in Hematology. So with all that verbiage, any other questions for Dr. Singh?
Hi guys, thought I would just chime in. That was a
Hi,
great
Karen.
overview, Pratibha. Thank you so much. So for people that know me, I'm Karen. I'm with the IU CCEH Center, and Pratibha's going to get the other lectures recorded as well. I think these are just a great series of lectures when you're starting with flow, because if you don't do what Pratibha has said, you can really get results that are not interpretable. So thank you for a great overview today.
Yeah, and standardization so that we can compare
Absolutely.
apples to apples or
Yeah.
at all research labs. Super important.
Yeah, those things are very, very important, especially for like when we are doing so complex multi-parameter rare population.
Right. Other questions? Okay, if not, we'll stop the seminar, and we'll remind you to keep your eyes peeled for looking for the posting of this talk. It will be found available on our center's CCEH.io website, but also on our YouTube channel, which we'll post at the top too, as soon as those recordings are processed and put online. And thanks again, Dr. Singh, for your presentation today.
Thank you.
Thank you.
Okay. Take care, everybody.
Should I stop?
So just what you'll do is you'll end the talk, and then it'll start telling you it's saving your recording. And it'll save it on your computer.
Okay. Where it is saying?
All you have to do is stop the seminar and it'll do it. There's no like question about save. It just starts saving the
Stop recording. Okay. Stop.
and then you shut